This lecture is on gene therapy. Gene therapy is a collection of techniques that change a person's DNA. It can be used for treating genetic disorders like sickle cell anemia or muscular dystrophy. It can be used for cancer treatments, inserting a gene that causes cancer cells to be more susceptible to a specific chemotherapy. It can be used to insert a functional gene into a patient who has a non-functional gene. It can add a new gene. Well, these are the dreams and the hopes. Let's talk about the reality. So I first want to talk about the so-called older technology gene therapy. And I say so-called older because this was really just first attempted in the 1990s. This technology uses a viral vector, which is a virus. It has been modified and it contains a corrected version of the gene that should be inserted. The virus that is most commonly used is what we call replication deficient. It's called adeno-associated virus, and the only way it reproduces in the wild is if it's in the same cell as an adenovirus. So it requires an adenovirus in order for it to replicate. If you just get this particular virus, the adeno-associated virus, nothing happens. And because it does not cause a disease, there's not a strong immune response to it. In addition, this virus has the ability to insert its DNA into a specific location on chromosome 19. There are a bunch of viruses that can insert their DNA into the host DNA, but they aren't usually very specific. And specificity helps reduce the risk that the inserted gene will end up being inserted in the middle of an oncogene, which can cause the growth of, oh, we know this one already. Onco is cancer, so that can trigger a case of cancer. This is an electron micrograph. Those little circles at the bottom that look like there's a bunch of things on the surface of them, those are the virus that we're talking about here, adeno-associated virus. So gene therapy has some risks. It can cause problems with the immune system. In particular, it can cause an autoimmune reaction where your immune system actually attacks your own body. It can end up inserting the gene in the wrong cell. If you need a gene in your liver, but the virus puts it in your lungs, that's not going to do you any good. You can use careful choice of injection site to reduce this risk. So neurological diseases, you can inject it into the inside of the skull, what's called an intracranial injection. I'd just as soon skip that. It doesn't sound pleasant. Or you can do the treatment ex vivo. Ex vivo means outside of the body. And this works really good for immune system problems or blood disorders because you can take stem cells out of the bone marrow in the top of the femur, treat those stem cells in the lab, and then return them to the top of the femur. This is it's not a very invasive procedure, and it limits it, or it puts the gene in the cells that need the gene. Okay, so we can reduce the risk of targeting the wrong cells. You could get an infection. I talked about how the most common vector does not tend to cause an infection. But if you've already got an infection with an adenovirus, well, then it can cause an issue. And other viruses have been tried, and those can cause infection, particularly in patients with weakened immune systems. And I mentioned already, there is a risk of cancer. If the new gene is inserted in the wrong place in your genome, this can cause the growth of a tumor, and of course that can progress to cancer. So that is the technology that has actually been tested in humans to this point. There's another technology, relatively new, that allows for much more specific gene editing. It's called CRISPR, C-R-I-S-P-R. This can be used to target a specific gene, and instead of inserting a healthy copy of the gene somewhere, this can actually go in and correct the mutations in the gene. So you're not inserting a whole new copy of the gene. You're actually correcting the mutation. This is still very early in research. The early work that we've been doing has not been as promising as it sounds. It shows that there is a risk of other genetic mutations going on in the same cells. So we need to understand more about why this is going wrong and how it should work, or how we can improve it before we actually try it in people. Both of these technologies are new. The idea of changing somebody's genes, well, that's not new, but the ability to do it is new. And since it does change a person's genes, it is currently only being used for more severe conditions. There's a whole bunch of cancer treatments that use gene therapy. There are several pretty nasty genetic disorders of the nervous system. There's a seizure disorder. There's a disorder that causes atrophy of the spinal muscles. It basically means the muscles shrivel and die. There's a genetic disorder that causes blindness. And there is a treatment for a condition called SCID, Severe Combined Immunodeficiency, which is a very, very severe malfunction of the immune system. People with this, if you're not careful and if you don't treat them for their immune dysfunction, they can die of just about anything that comes along, including something like the common cold. But we're changing people's genes. So there are ethical considerations. Are these modifications passed on to the next generation? Well, the current treatments do not get passed on because they are targeting specific cell types. And in order to get passed on to offspring, changes have to be made into the cells that end up producing the sperm and the eggs. Is gene therapy permanent? The treatments that have been approved so far are permanent, but there are some new techniques coming along that can make temporary changes. That's most likely to be useful in cancer treatments and not so much in genetic disorders. And then there's the idea of designer babies. So let me state right here at the beginning that if you go out on the internet and you look up information on this, you're going to find a lot of inaccuracies, myths, misconceptions, and fear-mongering. So, could you make designer babies? It would be done outside of the body, in the lab, before the embryo is attached to the uterine wall of the mother. So there's a period of about a week and a half between the time the egg is fertilized and it attaches to the wall of the uterus. And so that's the time period you would be looking at before the pregnancy really begins. So at this point, we can do what's called pre-implantation genetic diagnosis. If you know you have a genetic condition that is inherited or you're a carrier for it, you can have eggs from the mother, sperm from the father, combined in a lab, and then you can test those fertilized eggs, which are called a zygote, Z-Y-G-O-T-E, and see if they have that genetic defect. Do they have those mutations that would lead them to have sickle cell anemia or Tay-Sachs disease or cystic fibrosis? Pre-implantation genetic diagnosis can also be used for sex selection. And the laws about this vary dramatically between countries. So in theory, a zygote that is made in the lab could be genetically modified in the lab and then implanted into a woman. This has been done with other animals. Again, the laws vary dramatically from country to country. In 2018, a Chinese researcher claimed to have modified 2 zygotes, twin girls, that were born with a specific genetic mutation that makes them resistant to infection by HIV. Why he chose this one, it's not a life-threatening condition, nobody knows. Maybe he was just trying to show you could do it. But anyway, he claims to have done this. And of course, everybody got all upset about it. There was a lot of hullabaloo going on. As far as I know, those are the only true genetically modified babies that have ever been born. If there have been others born, it's been hush hush, and nobody's bragged about doing it. Now, while pre-pregnancy modification to reduce genetic disorders is a possibility, again, going back to these genetic disorders, you could get rid of the mutation that increases your to-be daughter's risk of developing breast cancer, for example. Changing other characteristics is much more challenging. Most human traits are multifactorial, and there are multiple genes involved. So if you're looking at things like athletic prowess, height, so-called intelligence, appearance, it's not as simple as changing one gene. And at this point, the technology to make these designer babies hasn't even been imagined yet. So that's just, we can theoretically correct inherited diseases if they're single gene diseases. But beyond that, not so much.